This is a lesson on plant tissues, and although it's stuck in with the gymnosperms, this is general information about all vascular plants. So ferns, gymnosperms, and angiosperms, this information applies. So you may find this helpful when you're looking at labeling your fern diagram, if you haven't done that yet, and also with the angiosperm lab that's coming up next. What we're listening to right now is someone who's created an optical record player that's actually playing the rings on a tree. So that's what you're hearing. If you're interested, the link is in the slideshow. There's a few other recordings of different species of trees. So quite interesting to hear. Now, the first thing we need to talk about when we talk about plant tissues is meristem. Meristem is like stem cells for plants. It is undifferentiated cells, so cells that haven't been directed to grow into a certain type of tissue, and they are what form the specialization of all other plant tissues. In plants, there are two different types of meristem. There's apical meristem, which occurs at the apex or the tip of the plant, so you'll find those at the tips of the shoots and the tips of the roots. It is clustered, and it's responsible for primary growth, so that's what makes the plants longer or taller. Roots go down, shoots go up. All plants have apical meristem. Now, the plants that get wider, the woody plants, those also have a layer of lateral meristem. And so lateral meristem is found in a cylinder around the edge of the plant, found throughout the length of all of the plant trunk, stems, shoots, branches, and roots. It can also be called the cambium, and it's found only in woody plants, plants that get wider from season to season. So here we can see these little clusters at the top. Axillary buds are the apical meristem at the shoots and the roots. And then we have these layers of lateral meristem, the cambium. And in fact, there are two layers. There's a vascular cambium and a cork cambium layer. So you can see those as we look through the different colors of tree rings where plant growth occurs to get wider. The vascular cambium is what lays down, that's the unspecialized cells that become the vascular tissues, the xylem and phloem. And then the cork cambium is what will become that outer layer of protective bark on the tree. So for woody growth or secondary growth, the wood part, right, the inside part that we would, use as lumber or recognize as, this is wood, that's made up of xylem. Remember that xylem is dead when it's mature, and it's hollow tubes that allow for water to move up and down. So that's why this is where we find all the sap hanging out in there. And then we have the bark, which is made out of a thin layer of phloem, the secondary phloem, and then periderm, which is another layer on the outside that is protective. So you can see here is our vascular cambium, and then we would have our cork cambium as well, in between our phloem and our periderm. If you're looking at a microscope slide, this is a fairly young tree. We can see 1, 2, 3 growth rings. It's the xylem where we're looking at these rings of growth. As the spring starts, water is plentiful, the sun starts shining, and so the tree grows very, very quickly in the spring. If you've ever seen little saplings poke up seemingly overnight and they grow very, very fast, those are those big, wide open xylem cells. They are typically a little bit lighter in color. So if you look at the differences in tree rings, the lighter ones closer to the middle are the early growth. And then as it gets later into the summer and cools off in the fall, water is less available in the summer, and it gets cooler and less sunlight in the fall. And so growth slows down, and those xylem elements are much narrower. And so we get a bit of a darker ring there. And that delineation allows us to count the rings in a tree and get the age. The outer edge here is what we would classify as the bark, and it's made up of the phloem and the cork cambium and then periderm, that outer protective layer. Remember that phloem is living and is responsible for bringing the materials from the leaves down throughout the rest of the plant to nourish the plant. So all that sugary liquid is traveling through the phloem. Little video about aging trees. And we'll talk about some leaves. So leaves are the primary site of photosynthesis. And although we're talking about gymnosperms right now, and these needles indeed are their leaves, these structures are true for all vascular plants, ferns, gymnosperms, and angiosperms. The leaf is a site where there's a potential large loss of water because there's a high surface area and a high rate of gas exchange. As gases move in and out of the leaf for photosynthesis, so can water. And so by changing the shape of the leaf, by coating it in a waxy cuticle, by changing the types of parenchyma cells, and utilizing the stomata and guard cells, we can actually help to minimize water loss based on leaf specializations. So typically broad-leafed plants do better in moist environments. As the environment gets drier, water is less available. We see leaves narrowing to needles or spines, or taking on, like the cactus, it's actually sort of fused together so that it can have water storage capabilities as well. We also see leaves where there's sort of a little fuzz or hair on it, and that has a few features. One of them is it doesn't feel very good in the mouth, and so it can help prevent predation. We know that those sort of fuzzy plants often deter predators or browsers or grazers, but they can also trap moisture, which can help the plant to absorb some of it through the leaves. And they also are aiding in minimizing the frost damage because it's the outer tips that kind of freeze first and can actually create a little insulating layer, almost like an igloo, over the plant to keep it warm. Now, if we're looking at a cross-section of a leaf, you can see the different layers of the leaf. So up here is the top surface, so what would be facing up towards the sun, and then down here is the lower surface facing towards the ground. First, outside the leaf cells, there is a waxy cuticle. So it's secreted by the epidermis, but the cuticle itself is not made of cells. Underneath that, we have a layer of epidermis. There is an upper and lower epidermis, and that is a layer of cells, and it contains the stomata, those openings. So there's guard cells in there as well. You can see a stomata down here with the two guard cells and then the air space in between. And then all of this inside stuff, the filler, is called parenchyma. This is where photosynthesis occurs, where the cells are able to repair, where materials can be stored. And so the two layers of parenchyma, we have the palisade layer or palisade parenchyma, where we have these column-like cells tightly packed together to maximize photosynthesis where the sun's going to be hitting the plant, so on the upper layer. On the bottom layer, we have a spongy parenchyma or spongy layer, where there's lots of air spaces to allow for gas exchange at the lower surface, where we're more likely to find the stomata. We've mentioned stomata before, but here they are on a scanning electron micrograph. You can see these solid cells are the epidermal cells, and then these things that look a little bit like coffee beans, well, those are two guard cells, and the opening between is a stomata. When water is plentiful and there's lots of sunlight, the guard cells swell up, which causes a little opening where gas can move in and out. And when water is not very plentiful or photosynthesis isn't occurring, then the guard cells shrivel, close up, and our stomata remain closed to trap any water in there. So that's it with our plant tissues. This information will help you out with the angiosperm lab as well at the end of this unit. So make sure you come back to this if you get stuck there.